Today we will talk about VDJ recombination. VDJ recombination is a process of making B and T cells specific. Before understanding VDJ recombination, let's look at B cells and T cells. B cells have B cell receptors, antibodies on their surface. Antibodies are Y-shaped and bind to antigens. Similarly, T cell receptors, TCR, exist on the surface of T cells and recognize antigenic peptides on MHC. The important thing is that one type of receptor can bind to only one type of antigen. Since there are countless types of antigens in the world, the same number of receptors are needed. Does this mean that there are countless genes to provide that many receptors? The answer is no. So how do they produce so many different types of antibodies and TCRs? Let's take B cells as an example. B cells start as lymphoid stem cells, then become pro-B cells, and finally become immature B cells. Pro-B cells are all the same, but during their development into immature B cells, they undergo VDJ recombination, which results in the production of different antibodies. Here is the explanation of VDJ recombination. The antibody is divided into a variable region and a constant region. The variable region consists of V segment, D segment, and J segment in the heavy chain, and V segment and J segment in the light chain. The combination of these 5 segments determines which antigen is bound. First, recombination occurs in the heavy chain. The original gene has several gene fragments for each of the V, D, and J segments, but they are actually used one at a time, with 65 different V segments, 27 different D segments, and 6 different J segments. There are 10,530 different heavy chains. Next, recombination takes place on the light chain. In the same way as with the heavy chain, about 200 different combinations of V and J segments are used to create the light chain. Considering the combinations of heavy and light chains, 2,000,000 and 6,000 different antibodies are produced. This process is called VDJ recombination. The diversity is further increased by somatic mutations. T cells do the same to produce many different types of TCRs. Where does this recombination take place? Both B cells and T cells differentiate from lymphoid stem cells, which reside in the bone marrow. B cells differentiate directly in the bone marrow, while T cells migrate to the thymus to differentiate. VDJ recombination occurs during differentiation. So recombination in B cells occurs in the bone marrow and in T cells in the thymus. In summary, VDJ recombination serves to create diverse receptors from few genes. This is all about VDJ recombination.